All right, good morning, everybody. Now, today's lecture is one of two, so it's a pair, where in both of these lectures we will be talking about what kind of chemical reactions we see in metabolism and what kind of enzymes, classes of enzymes, and what kind of cofactors these enzymes use for these specific reactions. So remember, in the very first lecture in this course, I was talking about the fact that metabolism is modular, so similar things repeat in different contexts. And one of the modules to speak about are the different types of reactions. So we'll talk about types of reactions, not specific reactions with specific molecules, but types of reactions, which you will see, or we will see together in many different metabolic contexts. And the hope is, once you understand the principle, then by looking at a specific metabolic pathway, it will be easier to understand why it happens this way. And it will also be easier to remember it, because once you understand the logic of a pathway, you don't have to memorize it, but you should be able to figure it out just from the logic of what goes on there. The other sense of modularity is related to cofactors, which are connected to specific reactions, because again, these repeat, okay, in different contexts. And once you know that a specific type of reaction is connected often with a specific cofactor, you don't have to memorize, okay, in this reaction there is this cofactor, because you know, okay, this type of reaction is usually catalyzed by an enzyme that contains this or that cofactor. So in these two lectures, we will go through types of reactions, types of enzymes, and types of cofactors that are linked to them, and take it as a preparation then for the metabolic pathways, because this should allow you to understand them better. Now, before we get to the specific reactions, let's just go through two or three general comments on metabolism, and I've already mentioned most of them, I think probably all three of them, but let's just revise them again. So one bit about terminology. Metabolic pathways or parts of metabolism are often divided into catabolic and anabolic. Okay, so what are catabolic pathways or reactions? Usually pathways. What are they? What do they do? How do we recognize them?
Break stuff down.
Correct. So they usually take a more complicated compound and break it down, sometimes all the way to carbon dioxide and water, and sometimes they produce some other things like ATP, which then we can use to power something else. On the other hand, anabolic pathways are building more complex compounds from simpler compounds. As I've said previously, for some metabolic pathways, this is really just a matter of direction. So we can use the same metabolic pathway in one direction as anabolic and in an opposite direction as catabolic. And again, we will see a few examples of that. For some pathways, we can't really say whether they are anabolic or catabolic because they're kind of doing both things. Okay, we will see those as well. But oftentimes we can tell whether this is an anabolic or catabolic pathway. The other thing, which again is a bit of a revision because we spoke about it, I think, yesterday, the idea that enzymes require some non-protein components which we call cofactors. And these cofactors can either be covalently or very strongly bound to the enzyme and they just remained in the enzyme, okay? Yeah, we call these prosthetic groups, okay? Even though, again, some people would say, well, an ion is not a prosthetic group, but let's call them all prosthetic groups. And then there's the other group of cofactors which only come for the reaction and they go away, and these are called co-substrates. Again, we'll see both of them today, and I will tell you which ones are which. The last thing I want to emphasize with these general comments is something I think I already mentioned previously, but let's just say that again. When we talked about thermodynamics and the laws of physics that tell us which processes can occur and which cannot occur, we all agreed, hopefully, that the second law of thermodynamics is telling us that only such processes which increase the entropy of the universe, or at least don't decrease it, are possible. And the ones that would decrease the entropy of the universe are impossible. They are thermodynamically forbidden. Now, that is all true, but locally, we can actually have processes that decrease entropy of this system, but they have to be coupled with some other reactions which increase the entropy of the universe by a larger amount. And this is the trick or the the mechanism why we can have actually anabolic pathways, why we can build complicated things, because we have to power them by some other process which releases a lot of heat into the surroundings and increases the entropy of the universe. One such process is, for example, the hydrolysis of ATP, right? Because ATP, as we spoke about, is kept far away from the equilibrium. So once we allow it to hydrolyze, it basically increases the entropy of the universe by a large amount. And that, if it is coupled to some other pathway or some other reaction which decreases the entropy, it actually powers it, okay? But it powers it by basically making sure that the entropy of the universe increases more, okay? General comments, again, something I think we spoke already about all three of them, but something to keep in mind in today's lecture, next lecture, and the lectures about metabolic pathway, specific metabolic pathways. Right. So let's start with the first type of reaction, which occurs pretty much everywhere. So whenever any kind of stuff is happening in metabolism, not literally everywhere, but most of the time. And it is a type of reaction which we usually disregard, okay? So in the metabolic pathways, we will almost never speak about this type of reaction, but it's almost everywhere. So it's quite important, but it's so simple that we don't talk about it. And the reaction that I'm talking about is dissociation. Dissociation, meaning a dissociation, for example, of a carboxylic acid, of a carboxyl group, or a dissociation of a protonated amine group. So often, these dissociations that we're interested in biochemistry are really movements of protons, either a binding of a proton, well, that would be association, not dissociation, right? That's the opposite direction, or releasing a proton from an acid. Now, if we have a carboxylic acid and it dissociates, what do we get? What are going to be the products? Sure. So we will get a carboxylate from carboxylic acid. We'll get carboxylate plus a proton. Okay. So this is, again, a very common thing that happens. And we can also have a similar reaction with amines or with some other basic groups. Let's have it like this so that it's in the same direction. So a protonated amine can dissociate into an unprotonated amine plus H+. Okay, and this works in both directions. So we see these acid-base reactions in as I say, in metabolism, we see them everywhere. Okay, almost every reaction will, or many reactions will involve this kind of dissociation. But we will usually not talk about them specifically, because in a way nothing interesting happens, right? It's just releasing and associating a proton, and there will be some equilibrium depending on the pH, how much the carboxylic acid is going to be protonated or how much the amine is going to be protonated or deprotonated. I will say, however, one thing which is related to metabolism as a whole, to human metabolism as a whole, and that is that if we take all the metabolic pathways occurring in the human body, overall, these metabolic processes tend to produce more protons than consume protons. So what we say is that our metabolism overall is proton productive. Or most pathways are proton productive. If we take the ones that consume protons and the ones that produce protons, there are more of those that produce protons. So this is just something to keep in mind when we talk about kidneys and some other organ systems later on. Just be aware that our metabolism tends to produce more protons than it consumes. So our bodies are actually set up in such a way that we're actually pretty good in excreting protons, okay? But we are not very good in keeping protons inside or making more protons, okay? So we are very good with dealing if there's too much protons, because that's what we're continuously producing. But we are not very good with dealing with situations when there are not enough protons, when we are too alkaline, okay? Now, I would just say another thing to that, because you may have come across these wellness and diet fads which talk about acidic foods and alkaline foods. Just raise your hand if you've heard of that, if you've come across. Okay, not many people, that's good, because it's not true, okay? We can't divide foods into those that produce more protons and that consume more protons. It's actually the metabolic pathways that produce them or not, okay? So this is not a good way of thinking about it, okay? But the truth is, and that is probably one of the bases for these kind of fads, is that really many of our metabolic pathways are producing protons, but as I say, our body is built to deal with it. Okay, so we don't have a problem with producing more protons. All right, that's dissociation, relatively simple. It's important because without dissociation, a lot of reactions would not occur, but it's so simple that we don't really talk about it very much. So, we can move to the next. Oh, and by the way, this dissociation doesn't require any enzymes, okay? It doesn't require any catalysis or anything like that, any cofactors. It just happens, okay? So that's also why we don't really need to spend too much time talking about that. So the next group of reactions is one which is much more complex and also interesting from the point of view of biochemistry. And those are reductions and oxidations. Okay, so redox reactions. Let's just recall together what is the principle behind oxidations and reductions? It's the movement of electrons. So oxidation is loss of electrons, so if something is oxidized, it's losing electrons to something else, and reduction is gaining of electrons. And remember, whenever we have oxidation, we must have reduction, okay? Because the electrons have to go somewhere, okay? So if we oxidize something, we have to reduce something at the same time, otherwise it wouldn't work. So we have redox reactions, which are, there are many, many, many in metabolism. We'll see many of them in different pathways. And these redox reactions tend to be catalyzed by the first class of enzymes, which are called oxidoreductases. Oxidoreductases, they catalyze these oxidation and reduction reactions. And we will go through two or three prototypical oxidation and reduction reactions, which we'll see repeat many different times in metabolism, and we will talk in detail about what is needed and how they work. So the first type of, in this case, oxidation reaction is the oxidation of alcohols into keto compounds. Yeah? You wanted to say into, okay. So if we take this kind of alcohol, we can oxidize it to, what will be the product of the oxidation? It will be an aldehyde, correct? Because it's a primary alcohol, right? It's a primary alcohol. So the product of oxidation will be aldehyde, an aldehyde. Sorry. Okay. Now, oftentimes, most of the time, this oxidation of an alcohol to an aldehyde in our metabolism is reversible. It tends to be reversible, okay? So we can usually take an aldehyde and reduce it to an alcohol, okay? Why am I telling you? Because then we'll see some other oxidations which are generally irreversible or very difficult to reverse, okay? But this one, generally speaking, is quite reversible. Now, since we said, we said this is oxidation. So oxidation means that the compound is losing electrons, is giving off some electrons. Now, the question is, how many electrons are lost by this alcohol in this oxidation?
2.
How do you know that it's 2?
Because NAD goes into NAD.
Well, we haven't really heard anything about NAD, but okay. Yeah.
There's 2 less H and each one has
Okay, so one way of looking at it is, okay, if we just take what atoms are disappearing or something like that, okay, there are 2 fewer atoms of hydrogen. Each of them contains 1 electron, so it is 2 electrons, okay? Or you could just calculate the oxidation state, the oxidation numbers, and you would see that really it is 2 electrons which are leaving this reaction. Now, I would slightly warn you against looking at hydrogen atoms, because there are reactions where hydrogen atoms leave in a way, but there is no oxidation, no reduction. They're not redox reactions. So I do understand how you figure it out. Okay, it makes sense. But be careful, because movement of hydrogens is not necessarily the same thing as movement of electrons. So be careful and really focus on electrons. Correct. So here it's 2 electrons that leave the compound. Now, since we said that any oxidation has to be accompanied by a reduction of something, then the question is, what is going to be reduced in this reaction? And very common partner in this reaction, the stuff that gets reduced, is a carrier of electrons called nicotinamide dinucleotide, or NAD+. Now, this is an enzyme cofactor, and in this reaction, this enzyme cofactor is a co-substrate, okay? Because NAD+ comes in, takes the 2 electrons, and reduces to NADH, okay? So it's literally a co-substrate. There's one substrate is this alcohol, and the other substrate is NAD+. They react together, and then they leave the enzyme. Okay, so this is a clear illustration what a co-substrate is. So NAD+ and its reduced form NADH are a very good example of co-substrates. They come in, take electrons, and leave. Now, I'm not saying that all oxidations of alcohols in our metabolism have to have NAD+ as the acceptor of electrons, but most of the ones that we will see actually do have NAD+ as the acceptor. So again, when talking about kind of general laws or modules or whatever, if you connect in your head an alcohol is being oxidized, probably NADH is going to be, or NAD+ is going to be involved. Okay? It's a good connection to have. It's not 100%, but close enough. Yes?
Is it gain 1 electron or 2 electrons?
2 electrons. So both electrons go onto NAD+. I'll show you in a second what actually occurs there, because oftentimes this is a bit mysterious. You know, how does it work? How does this compound accept these electrons? So let me show you how that works. So I'll draw the structure of NADH, or rather NAD+. You don't have to know the structure. No one will ask you to draw it, okay? But I think it's quite nice to see what it looks like to get an idea. So the interesting bit, the stuff where the redox reaction occurs, is the part of the molecule which is nicotinamide. So it's called nicotinamide adenine dinucleotide. And nicotinamide is where the stuff happens, where the electrons are being transferred. So this is what nicotinamide looks like. And it is bound to a sugar, to ribose. So sugar, ribose. Then there are some phosphate groups. Then there is another ribose there. And then there is adenine bound to it. I'm not going to draw the whole thing because it would take forever. So first of all, hopefully this explains why NAD+ has this weird name, nicotinamide adenine dinucleotide. What does it mean? Well, there is nicotinamide here, there is adenine here, and they form two nucleotides, because this is a nucleotide, and this is a nucleotide. Okay, because they contain a base, a sugar, and a phosphate, and this is what nucleotides need to contain. Okay, so nicotinamide, adenine, dinucleotide, hence the name. Okay, now, as I said, all the interesting stuff happens on the nicotinamide. The rest of it is just a carrier, okay? It's just there. No movement of electrons, no accepting electrons or anything like that. But what happens when you add 2 electrons to this nicotinamide is that the structure, and here notice that there's a positive charge here, okay? The structure changes in such a way that you can imagine that 1 electron kind of comes here. The double bonds rearrange, and we get 1 more hydrogen atom bound to the top. Now, I just drew 2 hydrogen atoms, but one of them was there all along, right? It wasn't drawn before, but it was there. Yeah? Hopefully, hopefully believe me that there was 1 hydrogen atom on it all along. Okay? But now I drew both of them so that you can see that there are actually 2 hydrogen atoms. So this is the change that occurs between NAD+ and NADH. This is NADH, and the stuff before was NAD+. So the 2 electrons, basically, one way of looking at it, you can say, well, it's the 2 electrons that form this bond with a proton, basically. I'll say that again, because now I see that it might be confusing. So what happens is, 2 electrons go in, they rearrange the double bonds and form bond or whatever, and then a proton comes in and completes this compound so that we have the hydrogen there as well. Why do I keep talking about this, what happens to hydrogen? The thing to focus on in these kinds of reactions is the movement of electrons. Not the movement of hydrogens, because that can be confusing, because there are situations when hydrogens move, but actually there's no net flow of electrons, okay? So always focus on electrons. The fact that then you need a proton to come in to actually form this new bond for hydrogen, right? That's true. But there are many protons around. There are always protons around, right? In any aqueous solution, loads of protons, right? No, maybe not loads, but enough that they can actually take part in these reactions. So the stuff to think about are electrons, the flow of electrons, and whether there are some protons coming in or not, that's not really that interesting, okay? Of course, it's interesting once you start studying the actual molecular mechanisms, but for us, the important thing is the flow of electrons. So 2 electrons will be accepted by NAD+, and it will get turned to NADH. Good. So for this kind of reaction, NADH is going to be oftentimes, or NAD+, the acceptor of electrons. There are exceptions, but mostly we'll see this. Any questions about this? No. Good. Now, as you all know, we can actually take the aldehyde and oxidize it further. And what do we oxidize it to? To a carboxylic acid, to carboxyl. Now here, I'm not really showing that it's reversible, because in most situations in our metabolism, it's not really reversible. Once you oxidize an aldehyde to carboxylic acid, it's very difficult to go back, okay? It's not completely impossible, but it's quite hard to actually do that. Now, how many electrons does it take to reduce an aldehyde to a carboxyl group? It takes 2 electrons. Again, if you calculate the oxidation states, you would see that. So it's 2 electrons that are released. And once again, the most commonly found acceptor for these electrons is NAD+. So it's going to be the same cofactor. Again, not 100%, but in most situations, it is going to be NAD+ that takes these 2 electrons and carries them somewhere else. And we'll talk about the pathways that work with NADH. Okay, now, very similarly, we can take a secondary alcohol, right? We can take a secondary alcohol and in a very similar way oxidize it reversibly to ketone. Once again, it's the same thing. So there are 2 electrons, and those 2 electrons usually go to NAD+. Okay? So it doesn't really matter whether it's a primary alcohol or secondary alcohol. We can just oxidize it by putting these electrons onto NAD+, making NADH, and we get a ketone. Now, can we oxidize ketone further?
You can, but it's very hard.
You can. It's quite hard, and it actually leads to disintegration of the molecule. Okay, so it is possible, but it usually means that the molecule has to break up. Okay, for further oxidation. There are situations like this in our metabolism, but generally speaking, these ketones we leave them alone. Okay, or we do something different with them. Okay, as we'll see in metabolism. So we can't directly oxidize it to carboxylic acid. Right. Okay. Any questions about this part of redox reactions? Good. So we can move on to the next type of oxidation. Still oxidation. We're still with redox reactions. But this time it is going to be an oxidation of a single bond in a compound, of a single bond between 2 carbons, into a double bond. So we have a compound like this, and we oxidize it, often reversibly, to this. Okay? Do you all see that it's an oxidation, that we are taking electrons away? Again, if you calculate the oxidation states, you would see that. It's one formal way of doing it. Now, how many electrons do we have to remove for this oxidation to occur? It's 2 electrons. Once again, it's 2 electrons. Okay? So you can see that so far there's quite an easy pattern. Okay? It's always 2 electrons. Oh, it's good to know. All right, so it's 2 electrons as well. However, most of the time, as a difference from the first type of oxidation that we saw, here we usually, in our metabolism, we usually do not use or cannot use NAD+ for accepting these electrons. Because, and this is a bit of a simplified explanation, so it's a bit simplified, basically NADH pulls in electrons with a certain force, with a certain affinity. It wants to get electrons, but its want to get electrons is not big enough to take electrons from a reaction like this. So for this kind of reaction, which is harder to do, we actually need some acceptor of electrons that has even higher affinity for electrons than NAD+. So we need basically a stronger oxidation agent. Okay? NAD+ is okay for this, but it would not be strong enough for this. So for these type of reactions, what we often use as an acceptor of electrons are derivatives of vitamin B2, which are called flavins. Flavins. They're called flavins because they are yellow. So from Latin, flavus means yellow. They are very yellow. Okay? And the 2 flavin cofactors that we often see in metabolism, most often see in our metabolism, are flavin adenine dinucleotide and flavin mononucleotide. Flavin adenine dinucleotide and flavin mononucleotide. So let's first talk about those names. The idea behind these names is the same as with NAD+. Okay? So in these compounds, we have flavin here. Then we have a sugar, phosphates, another sugar, and adenine. Okay? That's the case for FAD. So flavine, adenine, dinucleotide. Okay? With flavin mononucleotide, we only have this half of the molecule and not this half of the molecule. And it's just flavin mononucleotide. The other nucleotide is not there. Okay? Just the logic behind the names. In fact, if we want to be extremely pedantic, flavin adenine dinucleotide and flavin mononucleotide are not really correctly named, because the stuff to which the flavin connects is not really a sugar, but it's actually a sugar alcohol. It's a reduced sugar. It's not ribose, it's ribitol. So strictly speaking, flavin adenine dinucleotide should be flavin adenine mononucleotide, and flavin mononucleotide should be just flavin ribitol something, okay? So if we wanted to be extremely correct with these definitions. However, this is how they were named, so it doesn't really matter that it's not ribose, that it's ribitol, and we still call them these names, okay? But if somebody told you, careful, it can't be a nucleotide because there's not ribose, but there is ribitol, you can say, yeah, I know, but still, you know, it's still called these names, and we will use these names even though they are not, technically speaking, exactly correct. Now, I will not draw the structure of flavin. It's a bit more complicated, so I won't spend time with it. But the general idea of it accepting electrons and changing its structure is very similar to what we have here with NAD+. So the principle of what goes on there is about the same. However, the stoichiometry is a little bit different. So with NAD+, we have this plus charge here, so you can imagine that one of these electrons gets kind of consumed by the plus charge, okay? And the other one is needed for making a bond with hydrogen, okay? With FAD and FMN, we don't have any plus charge there, so we actually have to attach 2 atoms of hydrogen. So the reduced forms are FADH2, and with FMN, basically FMNH2. But we don't really see that much. It's the same thing. They're both accepting 2 electrons. But the stuff that happens to it, here we have 1 hydrogen added, and here we have 2 hydrogens added. But it's the same thing. It's just 2 electrons being transported. Now, here we come to the biggest difference between nicotinamide, or nicotinamide or NAD or nicotinamide-based coenzymes, cofactors, and flavins. And that is that while NAD plus and NADH are always co-substrates, so they come to the reaction and then they leave the reaction with these electrons, or come with electrons and leave without electrons, flavins are always prosthetic groups. They are always hidden inside an enzyme or hidden inside some protein or whatever. They are never just floating around, okay? That's a massive difference, and the reason why I'm stressing it so much is that many textbooks and diagrams in textbooks and schemes and whatever basically are wrong in this respect. They pretend as if FAD or FADH2 and FMN were just floating around. They're not floating around, they're always part of an enzyme or some other protein. I will tell you again when we talk about the specific pathways where this can be confusing, but just now get in your head one big difference between these two. So they're not different in how many electrons they take. They both take 2 electrons, okay? That's not a difference. The difference is this is a pure co-substrate, comes in and goes. This one is always hidden inside a protein. Usually the enzyme, or it could be some other kind of protein. Again, I will tell you once we see these examples. Now, I already started talking about vitamins, and I think I mentioned it in yesterday's lecture, that many cofactors are derivatives of vitamins or trace metals, okay, like copper and iron and what have you. So these two are also derivatives of vitamins. I already mentioned that flavins are derivatives of vitamin B2, which is called riboflavin. Okay, riboflavin, so you can see that these are derivatives of riboflavin. Nicotinamide, or NAD, is derivative of the vitamin nicotinamide. Sometimes I think it is denoted as B3. I don't really use this kind of notation for it. I think it's B3. But if you say that the vitamin is called nicotinamide, that's good enough. Or nicotinic acid, there are several different forms of it, okay? But nicotinamide is fine. So both of them are derivatives of vitamins. We can't synthesize them in our body. We have to take them in the diet. Good. Now I want to tell you about another type of oxidation reactions that we will see in our metabolism. Not as much as these. These will come in quite a lot, okay? But I want to tell you about another type of redox reaction that we can see, and another subclass of oxidoreductases which catalyze this different type of oxidation. And I want to talk about, are there any questions to this? Yes?
I did not understand why in this type of redox that we did, we don't use NAD+, but we use flavins.
Okay. That's a good question. Okay, I haven't really. I mean, I normally would have talked about it in the next lecture, but no. But that's fine. It's good that you mention it. So the question was about why do we use here NAD+ and not NADP+. There are, in fact, two versions of this coenzyme, and one of them contains a phosphate group bound here, okay, and that is called NADP+. It's the same thing. It does the same thing. It takes 2 electrons. It's basically exactly the same, with the only difference of this one phosphate group here. But, so it catalyzes similar types of reactions. It's, as I say, chemically speaking, it's almost identical. The difference between NADP+ and NAD+ is purely functional in our cells, in our metabolism. And the functional difference is that NAD+ is usually found in catabolic pathways. So pathways that break down stuff and take electrons from them in the process of breaking down stuff will use NAD+. For anabolic processes, where on the other hand we have to take electrons and give them to some process which builds more complex stuff, we're going to use NADP+. So even though chemically speaking they're almost identical, our cells have basically developed a mechanism for keeping those two processes apart. Because if you didn't keep them apart, you would have cycles, basically. So one pathway would give electrons to this, and another pathway would just take it away, and you would have these weird cycles. So the functional distinction between anabolism and catabolism is really mediated partly by the distinction in these cofactors. Okay? So in a catabolic pathways, we would use NAD+. In an anabolic, it's NADP+, but the chemistry is exactly the same. Okay? Yes? Yes.
And what is the FAD?
That's a prosthetic group. They're always covalently bound to a protein, to an enzyme, or something like that. Okay? Good. So, what is this next type of oxidation, or the next subtype of oxidoreductases that I want to talk about? Well, they are called oxygenases. Notice that for the oxidations that we just saw, the oxidation of an alcohol to an aldehyde into carboxylic group, or a single bond to a double bond, we didn't need any oxygen for that, right? We haven't used any oxygen. We just took electrons, put them on some carrier, and that was that. No oxygen was involved. But we do have reactions in our metabolism, oxidations, which do use oxygen. Often they use oxygen in order to add an OH group to a compound, so they're hydroxylases. They want to add a hydroxy group, okay? And for this reaction, they require oxygen because they are putting some oxygen into the compound. And these are called oxygenases, okay? Does it make sense? Right? They're adding an OH group, so they need to get this oxygen from somewhere, so they take in O2 as a part of the reaction mechanism. We'll see a few of those oxygenases, but let me just tell you that they can be divided into two subgroups. Monooxygenases and dioxygenases. As the name suggests, monooxygenases will only add one atom of oxygen to the molecule. So they will usually make one OH group. Dioxygenases will add both molecules of oxygen into the compound, usually as two OH groups, or there are some other possibilities as well. Okay? So the main distinction between monooxygenases and dioxygenases is how many atoms of oxygen they add to the substrate. Okay? It's relatively simple. We'll see a few of those. We'll see definitely a few monooxygenases. We won't see that many dioxygenases, I don't think, because the pathways where we have them, we never go into so much detail as to talk about those, but they do exist, okay? So be aware that there are redox enzymes which use oxygen. Most of them don't need oxygen. Some of them do. Usually it is to introduce OH groups into the compound. They're called oxygenases and can be divided into mono and dioxygenases depending on how many oxygens they add. Right. The final bit, and we'll have a short break after that, even though what I notice is that usually after the break the stuff stops recording. I don't know why, but we'll see. We'll try it again. Hopefully it's going to work even with the break. I think as people start moving around, it gets confused and it just switches itself off. So it's a bit annoying because then I have quite a lot of recordings now that only are the first half and the second half. But we'll have a break anyway. Before the break, let me just talk about a few more cofactors of oxidoreductases. So all of these enzymes, all of these that we just talked about, oxygenases, etc., apart from the two that we already talked about. So especially oxygenases tend to have as their cofactors, several different cofactors, but one of them that we often find there is heme. Heme, as you know, is a complex of iron. We have it in hemoglobin, but also in other types of enzymes and proteins, etc. So heme is a common cofactor, especially in oxygenases, but also in some other oxidoreductases. We'll see it in a few places. So that's another cofactor that we can associate with oxidoreductases. Now, heme, as you know, is not a vitamin, right? We make our own heme. The only thing that we require is the iron as a trace metal, so that we have to get from the diet, but it's not a vitamin. Heme is always a prosthetic group. It's always part of the protein, part of the enzyme. It's not just floating around, okay? It's always hidden inside some protein. It's a prosthetic group. Another cofactor, which is important for partly oxygenases, but also some other enzymes, has a really weird name. We talked about it actually in the very first lecture of this subject, okay, all the way back in October, but I'm sure you've forgotten about it. That's fine. It's called tetrahydrobiopterin. Tetrahydrobiopterin, tetrahydro is easy, right? 4 times hydrogen more, okay? That's easy, tetrahydro. But the compound itself is called biopterin, okay? It's some kind of a complicated system of rings, okay? It's called biopterin. We will only see tetrahydrobiopterin, I think, in 2 or 3 reactions in metabolism, not very many, but it's still worth knowing about because these reactions are quite important, okay? So that's another cofactor that we find in oxidoreductases. And once again, it is a prosthetic group. So it is bound to the enzyme, It never leaves the enzyme, okay? It's part of the enzyme. Quite important. The two trace metals which are connected to oxidoreductases are iron. We already spoke about iron. So iron is part of oxidoreductases either as heme iron, so iron which is in heme, but it's also possible to have enzymes which contain iron, but not as a part of heme. And this would be called non-heme iron containing enzymes, right? So I'm just putting iron here. And the structures in which this iron can exist, so one possibility is it's in heme, in this big ring, okay? But here we're talking about structures where there is iron, but not as a part of heme. And one of the possible structures, and we'll see it in a few enzymes, is called FeS clusters, or iron-sulfur clusters. Iron-sulfur clusters. And these are very interesting 3D structures where ions of iron are bound to atoms of sulfur, often in like a cube-like structure. So it doesn't have to be cube, but one possibility is a cube. So what you see in these enzymes is basically you have this big protein, and then in the center of the protein, you have this basically crystal of ferrous sulfide, okay, only like one crystal of ferrous sulfide, which is very important. It's in the active site and actually catalyzes some of these oxidoreduction reactions. Okay, so we'll talk about FeS clusters or iron-sulfur clusters as one possible cofactor. As you can see, it is a prosthetic group because it's covalently bound to the enzyme. Okay, this cube. The other trace metal connected to oxidoreductases is copper. And we will see a few oxidases or reductases that will contain copper inside. Once again, these iron and copper, we will talk about it a bit more in, I think, in three lectures from now or something like that, why we use these transition metals for these reactions. But let me just say, they are actually very handy when we want to move electrons around. These transition metals, their electronic structure makes it very easy. So that's why we have them in our enzymes. The final cofactor that I want to mention in connection to oxidoreductases is a very special co-substrate which we only find in one metabolic pathway, and it's called coenzyme Q. Coenzyme Q, another name for coenzyme Q is ubiquinone. Ubiquinone, and it is a co-substrate in the electron transport chain, in the mitochondrial respiratory chain. We will talk about it more there. Now, just raise your hand if you've heard of coenzyme Q. Many people probably have heard about it. Cool. So it's a co-substrate in one metabolic pathway in the electron transport chain. Just be aware now that it exists and that it's related to the function of some oxidoreductases. But we'll talk more about coenzyme Q when we talk about mitochondria. Good. Any questions? Yes?
I understand more or less the function of cofactors in these reactions that you talk about, but I was wondering if maybe you have a more specific definition for their role in every type of reaction.
Well, what these cofactors do, they take in electrons and move them somewhere else. So all of these things are basically transient carriers of electrons. That's their function.
This is only in redox reaction, right?
Well, yes, because when we're moving electrons, it's always a redox reaction, so yes.
I was wondering if cofactors of other types of reactions, maybe they have something in common about their function in all types of reactions.
Not really. So all of these are in oxidoreductases because they can take electrons and move them somewhere else, okay? When we talk about other types of reactions, we will see cofactors that can't do this, but they can do something else. They have some other chemical property that allows them to do something else. Okay? So the stuff that they have in common here is they are good acceptors or donors of electrons. Some of the other cofactors can't do this at all, but they do other things. Okay? But we'll see that as we go through other cofactors and reactions. Does it answer your question? Okay. We can talk about it afterwards if you want to specify it a bit more. Good. So let's risk a short break, okay? We'll see what happens with the recording. All right, so let's have a 4-minute break and we'll continue. Okay.
No problem. Say less.
So now we will move away from the first class of enzymes, oxidoreductases. So this whole thing was about oxidoreductases. Now we'll move to a different type of reaction, which is catalyzed by a different type of enzymes. And the type of reaction that I want to talk about is hydration and dehydration. It's the same thing, right? Just running in the opposite direction. So what is a hydration? Hydration is, in this context, the addition of a water molecule onto a double bond. Okay? That's what hydration means. So it doesn't mean drinking enough water. I mean, that's what it means as well. But in this respect, it means the addition of a water molecule onto a double bond. So what we have here is a double bond. We add a water molecule, and we get an alcohol. Okay? So this would be hydration. And in the opposite direction, it would be a dehydration. Again, a common reaction in metabolism. We'll see it in a few metabolic pathways. It's a relatively simple reaction, but quite important. And it is catalyzed by enzymes which belong to the fourth group called lyases. Now, you might think, why is it called a lyase? Well, or why does it belong? Why does it get classified as a lyase or as lyases? Can somebody try to answer that?
Because we're going to break the bond.
And which bond are we breaking?
CNO.
Correct. So the stuff that we are that we are breaking here is this bond. So the name lyase really applies to the opposite reaction, right? So when we look at it, here we're breaking OH, making a double bond. So that's a reaction for a lyase because it's breaking a bond. Okay? Well, not really, because it's not going to be ligase in the opposite direction, okay? Because we already have a classification. It's a lyase, okay? But it can run in both ways, okay? So it's still the same enzyme, but it can catalyze both directions, okay? But here it is called lyase because it's breaking this bond. Or, yeah, it's classified as lyase, not called lyase. It's classified as lyase. Now, these kind of reactions generally do not require any special cofactors, okay? So oftentimes the protein itself is enough to actually do this reaction, okay? So it's a relatively simple type of reaction. We'll see a few times. Just be aware of the name, what it means, what actually happens there, and what we classify it as. Before I have the question, I just want to focus your attention on the fact that this reaction is not a redox reaction. No electrons are leaving or coming in, okay? That's why I was a bit skeptical or careful about this idea that let's count hydrogens and that will tell us whether there's a redox reaction going on or not, okay? Because here, hydrogens are being added, right, to the molecule. But actually, it's not a redox reaction. Nothing gets oxidized, nothing gets reduced, okay? It's a non-redox reaction, okay? And if this is not clear to you, or if you're confused about that, really make sure you think about this, because it's important to know that this is not a redox reaction. Nothing is being oxidized. Okay? The redox state and the oxidation states of this compound are exactly the same as this compound. Yeah, sorry, you were first. But huh?
All the EC4 enzymes types.
No. Enzymes that catalyze this reaction, so hydrotases and dehydratases, belong to the group of lyases. But there are, of course, other lyases that do completely different things. Okay? Yep.
Why is it considered EC4 and not EC3? Like, why are they not hydrolases?
Oh, good question. Why is it not a hydrolase? Okay, EC3 hydrolases. Why is this not a hydrolase? Well, because hydrolases need water to break a bond. Okay? Here we are breaking a bond without needing any water. The water is actually a product of it. Okay? So hydrolases catalyze reactions where we take water molecule and use it to break a bond. Those are hydrolases. But here that's not what we're doing. We're actually breaking a bond without needing water to do it. Okay? So this is a lyase, not a hydrolase. Okay? Yes?
Reversible? It doesn't use.
It is reversible. Huh?
So because it is reversible, it doesn't actually need an addition of water.
No. So hydrolase is defined as an enzyme which catalyzes the breaking of a bond by using a molecule of water. Okay? Here we break a bond. That's true. This bond is being broken in this direction. But we don't need any water for that. Actually, the water comes out. So it can't be hydrolase. And that's true, but here we are not breaking a bond between two atoms. I know that we sort of are, but not for the definition. So here we're actually making a bond here. I know that you could say that you're breaking this one, but that's not what counts in this classification. So in this direction, you're actually making a bond, so it can't be hydrolase because hydrolase is breaking a bond. Okay? I know that this sounds a bit silly and kind of nitpicky. Okay? But that's what classifications do, okay? You have to be quite specific in what it means. So this is not going to be a hydrolase, it is a lyase. Yep? Good. Now, building on both of these types of reactions, so the ones that we had before, oxidizing alcohols and whatever, and the hydration and dehydration, we can actually put together a sequence of reactions which also occurs repeatedly in metabolism. And the sequence of reactions often is as follows. So we start with a compound that has a single bond between carbons. And we oxidize it to a double bond. How many electrons are taken away? 2. What is going to be the most likely acceptor for these electrons? FAD. FAD could be FMN, but usually it's FAD, okay? Because NAD is not strong enough to take these electrons from it, okay? I'm just kind of revising what we talked about in the beginning. So 2 electrons go away here. In the next step, we hydrate the double bond, which is not a redox reaction. No electrons are going in, no electrons are flowing out. Not a redox reaction, and we get an alcohol. We get an alcohol from it. And then we can oxidize this alcohol. Now, of course, if it's a tertiary alcohol, we can't do that, but it's not a tertiary alcohol in this case. So we can then oxidize it and get a ketone. This is a redox reaction, so 2 electrons will be taken from it, and the most likely acceptor for these 2 electrons is going to be NAD+, absolutely. Okay? So this sequence of reactions, again, is something that we will see in metabolism a few times, okay? So it's worth seeing it. This is a redox reaction, this is an oxidation, this is not a redox reaction. This is a redox reaction, oxidation. Okay, so this is not a new type of reaction. I just connected the reactions that we already talked about into a sequence which repeats in metabolism. And now you see it here. Hopefully you will be able to identify it in the metabolic pathways themselves. Now, the third and last type of reactions and group of enzymes that we'll talk about today are isomerizations. Isomerizations. Now, as the name suggests, these reactions convert one isomer into another. The enzymes that catalyze these reactions are called isomerases. And they have their own class, EC5. And we will see in the metabolic pathways that we talk about, we will see quite a lot of isomerases. So it's quite a common thing that we need to convert one isomer to another. However, to complicate things a little bit, as there are different kinds of isomerisms, we can have different types of isomers, right? So we can also have different types of reactions, isomerization reactions, and we can also have different types of enzymes that catalyze these different kinds of isomerizations, okay? So, and they have, sometimes they have names which are a bit weird and they don't really explain what they're doing, and that's why we have to talk about them in this lecture and really specify what they do. So, the first type of isomerism that we often need to change is optical isomerism. That is, we have a chiral, usually carbon, okay, so we have a chiral atom, and we want to change the configuration on the chiral atom, okay? Change the optical configuration. Now, the reaction is just one, okay? So we're changing the optical configuration on a chiral atom. But we have actually two different kinds of enzymes that can do it, okay? It's weird, but that's how it is. So the first type of enzymes that can catalyze this optical isomerization are called racemases. Because a racemate, does anyone know what it is? Or racemic mixture? Correct. So a racemic mixture is where both optical isomers are present. Okay? So you could say that racemase is creating a racemic mixture out of two isomers or whatever. Okay? I know it's a weird name, but that's what it's called. Okay? A racemase. The other type are called epimerases. Epimerases. Now, what's the difference? As I said, both of these enzymes, what they do is they change the optical configuration on a chiral atom. But the difference is that racemases do this in molecules where there is only one chiral center. Epimerases do it in molecules where there's more than one chiral center. They will still only change one atom, okay, at a time. But if the molecule has only one, they're usually called racemases, okay? If there are more than one, they're called epimerases. We will see quite a few of those. With the epimerases, usually it is in the metabolism of carbohydrates, of saccharides, because saccharides will have quite a few chiral centers, okay? And if we flip just one of them, we need an epimerase for that. Racemases are for simpler compounds where there's only one chiral center. So that's for optical isomerism. Now, the other type of isomerism, yes?
Can we also call racemases mutases?
Well, that's a bit confusing because historically speaking, mutases generally are doing something slightly different, okay? And I'm actually coming to that, okay? So now we have structural isomerism, and structural isomerism basically means where in the molecule are the functional groups, right? So if we move them around, we create a structural isomer. And this is what is often catalyzed by mutases. So mutases are enzymes that can move functional groups around the molecule. Okay? So they will cause structural isomerism. They just cut off a group from one point of the molecule, from one side of the molecule, and move it to another. Now, do not confuse these enzymes with transferases, which is a separate group, separate class, because transferases will transport or will transfer functional groups between different molecules. Here we are moving within one molecule. Okay? The next group of, or the next type of isomerism and corresponding group of enzymes is geometric isomerism, so cis-trans, right? The enzymes that catalyze this are called cis-trans isomerases, so nothing, no special name. Cis-trans isomerases. And we have actually very few of those in metabolism, and we will really not talk about any of those in metabolism. But I just want to mention one very important cis-trans isomerase, which is important for the synthesis or for the function of our proteins, and that is called peptidyl prolyl isomerase. I'll explain what that means. So, peptidyl prolyl isomerase. So when we build a protein, and the protein contains in its chain the amino acid proline, hopefully you will all recall that proline is a weird amino acid, because the amino group is actually part of a ring, right? So imagine that you're adding more and more amino acids, and then you put in the ring. And there are two different ways of continuing from the ring. Either the peptide can go under the ring and continue, or it can go above the ring and continue. And this is a cis-trans, that's a geometric isomerism. Now, once we build the enzyme, we have to be able to flip this around, otherwise the enzyme or the protein that we're building might not fold properly, because we have to be able to fold it, right? So these peptidyl prolyl isomerases, what they do is they're ready, and if we need to flip the configuration in folding the protein, that's what they do, okay? So it's a very special type of enzyme. It's a group of enzymes that do it, and they are very important for protein folding, okay? You will never hear about cis-trans isomerases or anything like that probably ever again, but just be aware that these peptidyl prolyl isomerases are super important for making proteins or making them functional in our cells. The final type of isomerism, well, actually, it's also structural isomerism, so I will just continue kind of here. Another subtype of isomerization reaction, or rather class of enzymes that do it, are, and this is, you're going to hate me because this is going to be really confusing, they are called intramolecular oxidoreductases. Now, the first question you might have, well, why aren't those under oxidoreductases? Because in these reactions, we oxidize one part of the same molecule and reduce another part of the same molecule. So the electrons never leave the molecule. They're just kind of moving around the molecule, okay? So these are not considered as oxidoreductases because we're not removing or adding any electrons to the compound. We're just switching them from place to place. Okay, so intramolecular oxidoreductases are really just a type of isomerase. And one reaction that we will see in our metabolism, which is actually catalyzed by an intramolecular oxidoreductase, is the isomerization of an aldose into a ketose. In sugars, right, we have aldoses and ketoses. And if we want to isomerize an aldose into a ketose, what we do is we reduce this one and oxidize this one. So it's an intramolecular redox reaction, intramolecular oxidation reduction, okay? But it's really just isomerization. So it's not, the electrons are not leaving the molecule. We just kind of move them around, okay? So this would be a type of reaction catalyzed by an intramolecular oxidoreductase. Good. That's all I wanted to talk about. So if there are any questions, we have time to answer them. Yes?
Are race maces the same ones that we use in pharmaceuticals?
Say again?
Race maces, are they the same ones they use in pharmaceuticals?
So the question is whether racemase are used in pharmaceuticals. It's just a confusion about terminology, okay? So a racemase is an enzyme that flips the configuration. A racemate is a mixture of two optical isomers. And in pharmaceuticals, you can either have a pure isomer or you can have a racemate. But it's not a racemase, it's a racemate. So racemate is a mixture of two optical isomers. Racemase is an enzyme that flips the configuration.
So when, like for example, thalidomide, one of them causes like limb damage.
Correct, yes.
So we only need to use one of the, so we use race maces for that, or no?
Usually we just use chemical methods to do it. You wouldn't use like biotechnological methods. It's possible in theory, but not for this. Any other questions? Any wish to clarify something? Nobody wants me to clarify anything. I get it. All right. We'll continue next week with the second part of this lecture, and then we'll start the metabolic pathways proper. Right. Yes, that was just half of it.